Have you ever stared at a science investigation question and just your mind goes blank? You know that feeling, where do I even begin? Well, that's exactly what we're going to tackle today. We're going to turn that big, scary question into a simple, step-by-step plan. So if this question right here feels a little too real, hey, you're in the right place. It's a super common feeling, but trust me, by the end of this, you are going to have a rock-solid framework to plan out any investigation with total confidence. All right, so every great plan starts with knowing what tools you have in your toolbox. And in biology, one of your most important toolkits is the set of food tests. So let's do a quick fire round just to make sure we've got these down cold. First up, this starch test. This one is beautifully simple. You just take your food sample, add a couple of drops of iodine solution, and that's literally it. Now you just wait and see what happens. And what are we looking for? This really dramatic color change. If that yellowish-brown iodine solution suddenly turns a deep, dark blue-black, boom, you've got starch. If it just stays that yellow-brown color, then there's no starch. Super clear, right? Okay, next up, reducing sugars, things like glucose. Now this test has an extra step. You add the Blue Benedict solution, but then, and this is key, it needs some heat to get the reaction going. And the safest, most standard way to do that is in a hot water bath. Now, here's the really cool part about the Benedict's test. The result isn't just a simple yes or no. If you've got sugar, that blue solution will change color, and it actually creates a whole spectrum. It can go from green to yellow to orange, all the way to a brick red. And the further along that color spectrum you go, the more sugar there is. If it stays blue, you've got nothing. Okay, let's finish off our toolkit with a few more essentials. For protein, you're going to add burette reagent and look for a nice purple or lilac color. For fats, you'll do the ethanol emulsion test, and if you see a milky, cloudy layer form, that's your positive result. And finally, for vitamin C, you add the blue DCPIP solution, and if vitamin C is present, it'll go completely colorless. And just like that, you have a whole suite of tests ready to go. And here it is, all laid out for you in one place, your food test cheat sheet. Seriously, this is an amazing summary for revision. It's got the nutrients, the chemical you need, and the color you're looking for. It's a super handy reference. Okay, so we've got our tools. Now let's get into the logic of the experiment, which is all about the variables. Honestly, getting your head around these is probably the most important part of designing a great investigation. Let's start with the star of the show, the independent variable. This is the one and only thing that you are choosing to change in your experiment. So if you're testing how temperature affects an enzyme, temperature is your independent variable. If you're testing different light intensities on a plant, light intensity is your independent variable. It's the whole point of your experiment. So if you change something, you obviously need to measure what happens, right? Well, that's your dependent variable. The result you measure depends on what you changed. See, it's right there in the name. This is what you're actually recording. Maybe it's how much gas is produced or how long a color change takes. It's your result. Now listen up, because this is where so many experiments fall apart. The controlled variables. These are all the other things that could mess with your results, so you have to lock them down and keep them exactly the same. We're talking about using the same volume of liquids, the same concentration, the same species of plant, everything you're not testing. This is what makes it a fair test. It's absolutely crucial. Okay, let's clear up something that trips a lot of people up. The difference between a controlled variable and a control experiment. A controlled variable is just one factor you keep constant, like temperature. A control experiment is a whole separate test you run where you take out the independent variable completely. Why? It gives you a baseline, and it proves that the thing you're changing is what's actually causing the result and not something else. So, we have our tools. We get the logic. Now it's time to put it all together. Let's build a flawless, step-by-step plan that you can literally use for any investigation that comes your way. So, how on earth do you remember all the important bits and pieces of a good plan? Easy. With this one, slightly strange but incredibly powerful mnemonic. DRSCICT. I know, it sounds a little weird, but I promise you this is going to be your secret weapon. So what does this weird phrase even mean? Well, the D is for your dependent variable. R is for repeating your experiment. S is for safety precautions. Then you've got I for your independent variable, C for your controlled variables, and T for your tools and the time you'll run it for. If you can just remember DRSICT, you will never forget a key part of your method. And here is how you put that into action. Think of this as your recipe for a perfect plan. First, state your independent variable. and try to have at least 5 different values. Next, list out all your controlled variables. Then, write out your method step by step, mentioning the tools and the time. After that, be really specific about what your dependent variable is and how you'll measure it. And finally, the golden rule. State that you'll repeat the whole thing at least 3 times and get an average. Follow these 5 steps, and you're golden.
A great plan is one thing, but making it happen in the lab is another. So let's talk about leveling up your practical skills by looking at some of the most common mistakes people make, and more importantly, how you can sidestep them completely.
All right, let's look at some classic lab errors. Judging color by eye? That can be pretty subjective, right? A colorimeter gives you an objective number. Squeezing drops from a bottle? The drops are never the same size. A pipette gives you an exact volume every single time. Not mixing things the same way each time? That can change reaction rates. A magnetic stir makes it perfectly consistent. These little upgrades make a huge difference to how accurate and reliable your data is. And here's a perfect example. For a test like the vitamin C one, where you're adding one liquid to another until you see a color change, that's called a titration. And using a burette, this piece of equipment here, is a total game changer. It lets you add the liquid with incredible control, literally 1 drop at a time. It completely gets rid of the error from uneven drops and makes your results so much more precise. Okay, you've planned it, you've done the experiment, you've collected all your numbers. So now what? The final piece of the puzzle is to present that data in a way that's clear, professional, and actually tells the story of what you found. Everything starts with a good, clean table. This is the foundation. Keep it simple. Your independent variable goes in the first column, and your dependent variable goes in the second. But here's the pro tip that so many people forget. Always put the units in the column headings, not next to every number. It's a small detail that just makes everything look so much more professional. Okay, now it's time to turn those numbers into a picture. But which graph do you use? It's actually pretty simple. If your independent variable is in separate categories, like types of fruit, you use a bar chart. If your data is grouped into ranges, like height ranges, you use a histogram. But for most experiments, where your independent variable is continuous, like temperature or time, a line graph is going to be your best friend. And finally, never forget that everything you produce is data, even a biological drawing. It needs the same amount of care and precision as your tables and graphs. So use clear, unbroken lines and label everything accurately. You see, a successful investigation isn't really about one single moment of genius. It's about a whole series of small, careful, well-planned steps that all add up. Now that you have the tools, what are you going to go investigate?